We conclude the foundation modules with a look at genetic markers, a topic that, like the others previously covered, could embrace a wide range of material and take up hundreds of slides. In our constrained treatment, we will focus largely on a specific type of genetic marker, the SNP, or single nucleotide polymorphism, following a general introduction. To say that markers are important to the science of genetics is an understatement. Not only have they provided the tools to investigate genetic mechanisms, but they are the means by which we endeavor to dissect complex genetic traits and complement traditional crop improvement methods. We have noted in previous modules that polymorphisms may take any of many forms, from insertions or deletions of one to many bases in a stretch of DNA to the simplest of mutations, the replacement of one base with another, called a SNP or single nucleotide polymorphism. When the polymorphism can be detected using any of the myriad tools available today, and it segregates in a Mendelian manner, it represents a useful marker. This, of course, is key to markers that originate in the nucleus. An individual may be homozygous for either allele, or heterozygous, that is, have one copy of each allele, for such markers. Organelle genomes are almost exclusively inherited uniparentally. Polymorphisms in organelle genomes are therefore detectable only among individuals, assuming they are allelic. Both nuclear and organelle markers find application in forestry, though we will mainly focus on the former in coming modules. The science of genetics would not have been possible without mutations detectable as markers. Think of Mendel's peas and the 7 or so morphological markers that led to the understanding of concepts such as genes and segregation. Consider the concepts of crossing over and recombination, and how markers elucidated these phenomena. The search for new marker classes has been ongoing across a spectrum of scientific disciplines since the rediscovery of Mendel's work, and reached a frenzied pace in recent decades with the introduction of new and better technologies. The development of markers in forest trees has generally followed technology improvements made by those studying humans, model species, and ultimately, crop species. Adoption of new marker technologies has often been fast, though cost hurdles have often prevented widespread application. The development of protein electrophoresis in the 70s permitted the rapid and inexpensive detection of dozens of allozymes, gene products, and opened the door to all kinds of studies, from conservation and population genetics to paternity analysis and genetic integrity applications. Allozymes were followed by the dizzyingly fast-paced development of other marker types that found immediate, if sometimes short-lived, applications in tree breeding and basic genetics. Some of these, such as allozymes and SSRs, still find specific utility, though by and large, most genome-scale science is now reliant on the SNP. Molecular genetic markers clearly changed the face of forest genetics from the scope and scale of research being conducted to the types of students who seek postgraduate studies in the field. Though markers offer great promise to practical tree breeders, promises now made for over 25 years, they have not replaced traditional tree breeders yet. Sadly, however, the training of traditional tree breeders, or breeders of all crops for that matter, has suffered because of the sustained support for molecular genetics studies by granting agencies. It is an interesting conundrum. As the cost of markers declines and the ease of use has increased, marker applications have blossomed. We will elaborate on many of these applications at some length in later modules, but a quick accounting here should give the viewer a sense that markers have found a place in forestry. Simply being able to unambiguously say that a particular tree, or portion thereof, is what is believed to be, via genetic fingerprinting, represents a financial and potentially legal boon to plant breeders. Markers can make tree breeding faster, less expensive, and deliver greater gain than traditional methods, either by simply improving management techniques or by assisting the selection process. Gene resource managers can rely on markers for guidance on gene conservation practices to restore restoration of degraded environments to assisted migration efforts. Each type of marker has a set of characteristics which dictate its utility. Within marker types, there is typically variation among individual markers in their usefulness. Thus, there are a number of things to consider when selecting a suite of markers for a project. First off, the marker must be interpretable and reliable. What does that mean? To be reliable, it must show up as a distinctive marker in all tissues and at all times in the organism's life cycle. Not necessarily true of all allozymes, for instance. To be interpretable means, mostly, that the patterns of polymorphism observed, whether as blots on a gel or fragment sizes on a sequencer, are segregating in a Mendelian manner. Markers that have one allele that occurs very frequently, that is, greater than 95% of the time, may not be too useful for some applications, like paternity testing. Conversely, markers with multiple alleles, each of nearly equal frequency, can be very powerful for such applications. The microsatellite, or SSR, is a good example of such a marker. However, SSRs typically are expensive to find and develop, and generally do not occur in large numbers for a given taxa. For applications like genetic mapping, where several hundred or more markers may be needed, an alternative like rapids or SNPs would be desirable. A major factor to consider is cost, and this has multiple components. Namely, what is the cost of an individual data point? That is, one marker for one individual, how many markers are needed for the application, and how many individuals need to be genotyped. Finally, though not entirely separate from cost, are issues related to technology. What types of technology platforms exist for your chosen markers? Can you do the work internally? That is, have trained personnel and equipment, or must it be contracted out, and so forth. The general trends in recent years have been to outsourcing marker work, that is, genotyping, to contract labs that can operate multiple technology platforms in high-throughput processes that result in relatively low costs to the client. The cost of genotyping SNPs, for example, has dropped to mere pennies per marker and less, depending on the platform, though for small projects requiring just a few markers, let's say less than 96, costs more in the range of $0.25 per marker are common. This is a stark contrast to costs that typically were in the $3 to $10 per marker range 10 to 15 years ago. Over the next few slides, we will introduce three types of markers: Allozymes, SSRs, and SNPs, the first two of which are mentioned only briefly, for they still find utility in forestry applications. SNPs will be discussed at length. Allozyme markers are genetic polymorphisms within a gene that have resulted in differences in amino acid constitution and protein configuration in the protein encoded by that gene. They are detectable because the protein forms possess different electrical charges and separate in space within a matrix, such as starch or acrylamide gel, when put under electric field. Detection depends on preserving biological enzyme activity of samples prior to electrophoresis. Protein mobility is detected as a colored band, which appears upon staining on a starch gel slide. Sample processing is typically minimal. Crude extracts from seeds or needles, relatively fast and inexpensive. Lab infrastructure and chemical and equipment costs are also relatively minimal. From the photo above, we can illustrate several points. There are 15 columns and basically 3 rows of information shown here. The columns represent samples of individual trees or seeds. The rows represent, ostensibly, the products of 3 different genes. Our interpretation here is that the 2 bottom rows represent gene products that are homozygous among these samples. The top row shows patterns that suggest individual diploid samples are homozygous for either a fast mobility variant, samples 2, 6, and 8 through 14, or a slow mobility variant, samples 4 and 5, or they are heterozygous for both variants, samples 1, 3, 7, and 15. One can easily calculate genotype and allele frequencies from this type of data. You can also see, from the differences in intensity of bands, that reliability of this type of marker may occasionally be suspect. Allozymes have been used extensively for comparative studies across taxa of all life forms. Allozyme loci generally possess 2 to several alleles, though often one occurs much more frequently than the others. Most alleles are visible, though occasionally a null allele occurs and requires pedigree crosses to detect. For conifers that possess seeds with megagametophytic tissue, that is, endosperm and flowering plants, derived from the mother and embryo tissue derived from the union of male and female gametes, it is easy to use allozymes to deduce the genotypes of male and female parents by running the 2 tissues side by side. While powerful tools for their time, allozyme shortcomings have prevented their expanded use, especially for genomic applications that require large numbers of markers and individuals. With the advent of polymerase chain reaction, or PCR, technology, a wide array of marker types were developed. Among these, the microsatellite, or simple sequence repeat, that is, SSR, has gained widespread application in many organisms, including forest trees. SSRs are particularly useful for fingerprinting and paternity analyses because of their codominant multi-allelic nature. The SSR marker polymorphism consists of errors in DNA replication that result in short runs of 2 or more bases being repeated several times, one after another. In the example shown here, the repeat unit is A, T, C. One allele has 7 repeats, and one has 8 repeats. In a population of trees, individuals, there may exist many alleles, 2 to 40 or more. The figure at the bottom of the slide illustrates the genotypes of an array of unrelated individuals. These diploid genotypes exhibit 4 alleles at one locus. Some individuals are homozygous, only 1 allele is showing, but most have 2 alleles. That is, they are heterozygous, which illustrates the high information content of this type of marker. SSRs are the marker of choice for human paternity analyses. While SSR markers are very useful for some applications, their utility is ultimately constrained by the cost and difficulty of developing and using them. Though they are numerous, finding them in genomic sequence is difficult, and converting them to good markers is time-consuming and costly. Generally, a rather small percentage of potential markers are converted. A common problem is finding good primer sites on either side of the repeat units. Mutations in these areas commonly result in null alleles, even for markers that have converted. Finally, the cost of genotyping, that is, $1 to $8 per marker per individual, is prohibitive for large-scale applications. The third marker we will discuss is the single nucleotide polymorphism, or SNP. The SNP is the most common type of polymorphism found in the genome of virtually every organism studied. Though their frequency varies a great deal within the genome, that is, by chromosome, chromosome region, and gene, and among species, most higher plants and animals possess literally millions of SNPs. SNPs occur with an average frequency of about 0.1%, that is, one SNP every 1,000 bases, with some regions having as high a frequency of 4 to 5%. The SNP example shown in this slide is illustrated by a 2-allele, 3-genotype locus within a short DNA sequence. SNPs are useful as genetic markers because of their abundance in the genome, relatively unambiguous assay techniques, that is, accuracy of detection, slow mutation rate, and low genotyping costs. They are not particularly useful for comparative studies among unrelated or distantly related taxa, however. We will spend the remaining time in this module discussing the SNP, its properties, methods of discovery and detection, and processing to make useful markers. The bases, or nucleotides, that make up the DNA code are classified as either purines or pyrimidines. Bases always pair with a specific member of the opposite class, A with T and G with C. SNPs may be classified as being either transitions, that is, a change to the other base in the same class, or transversions, that is, a change to a base of a different class. SNPs occur everywhere in the genome. For instance, SNPs may occur in non-coding intergenic DNA regions or within genes, or in regions that are neither transcribed, for example, enhancers, promoters, and introns, nor translated, for example, UTRs, or untranslated regions. SNPs in any of these regions may affect the gene product. SNPs encoding DNA, for example, within the exons that code for amino acids, may be classified as synonymous, non-synonymous, or nonsense. The first represents a SNP at a position that does not result in an amino acid change. Recall that the redundant nature of the genetic code, especially at the third position. Non-synonymous SNPs result in amino acid changes and consequently protein changes, and are most likely to result in changes in fitness. A SNP that causes a change in phenotype of the organism that is detectable is often called a QTN, or quantitative trait nucleotide. Conceptually, SNP discovery is straightforward. Obtain genetic sequence of the same chromosomal region from 2 or more chromosomes, align them so that the base positions are identical for all sequences, and look for differences among the sequences at each position. It sounds simple, but in fact can be quite difficult to do well. If you were to start from scratch specifically to find SNPs for a given organism, you might follow one of 3 approaches as noted here. Number 1, look for existing genetic sequence online, and use available bioinformatic tools to identify the SNPs, or look for online SNP databases where they have already been identified. 2, undertake resequencing of a targeted region, for example, a known gene for which you have a sequence already. Or 3, conduct de novo sequencing. The amount of sequence processing necessary to identify and verify SNPs for these different sources will vary. We will discuss these steps in future slides. Finally, once a SNP is detected and verified, methods must be identified to genotype your population for that SNP, and most likely hundreds of other SNPs, preferably all at the same time. Finding the right technology platform for your application may require considerable study and some compromise. For those seeking to find SNPs for species they are working with, a quick search of electronic databases may reveal what they seek. A number of large tree genomics projects are typically underway in the US, Canada, and abroad. Of course, the Clearinghouse for Genomic Information in the US, that is, the National Center for Biotechnology Information, or NCBI, maintains sequence and marker files for all species ever studied using government funding. Electronic searches are not always easy, but are fast, inexpensive, and may be very rewarding. The downside to such data is that results rely on existing sequence data of variable quality, reflected by a high false positive rate of SNP identification. However, newer data are increasingly of high quality, and tools exist to evaluate SNP quality when sequencing is available for study. Before the development of second-generation sequencing tools, in approximately 2005, a common approach to obtaining high-quality SNP data was to use Sanger sequencing methods to obtain a targeted chromosomal region or known gene for each of several individuals and to compare their sequences after alignment. Candidate regions chosen for sequencing were typically selected based on known function of the gene being targeted, obtained from other sources, or unknown genes obtained from EST library construction. The approach is still valid, though rather more costly and time-consuming than high-throughput sequencing available today. A big advantage to this approach is that SNPs that occur at low frequency in a population are easily found, and allele frequencies can be calculated. Outgroups can be easily compared. This slide provides a detailed look at a resequencing approach to SNP discovery in Loblolly pine. It began by collecting all known EST contig sequence information for the species. ESTs, or expressed sequence tags, are sequences that represent some portion of an expressed gene. They may be anywhere from 100 to 1,000 base pairs or more in length. These are obtained from sequencing complementary or cDNA obtained from mRNA or messenger RNA collected in specified tissues of the host organism. In the first step of the process, software was used to filter the original list of putative contigs or unigenes down to a more reliable list of potential candidate genes, approximately 20,000. Primers were successfully developed for approximately 14,000 of the putative unigenes using automated, high-throughput primer design software. These primers were synthesized and used in a test sequencing of a few individuals to determine if they worked properly. Of the 14,000 tested, approximately 7,900 SNPs were successfully synthesized. That is, of the 14,000 tested, approximately 7,900 ESTs were successfully synthesized. These validated primers were subsequently used to sequence a panel of 24 individual trees using genomic DNA. The trees were selected to represent a cross-section of the species range. From this database, some 40,000 SNPs were detected, ranging in frequency for the most common allele from 50% to 96%. The higher the number of individuals, or more accurately, the higher the number of chromosomes sequenced, the more likely it is that low-frequency alleles will be discovered. Alleles may be infrequent in the population because they are relatively new and only just becoming established, i.e., that are selectively advantageous or increasing by chance due to founder's effects, etc. Or they may be rare because they are deleterious. Gene conservation programs tend to favor maintenance of low-frequency alleles to retain as much variability as possible. Allelic breeding values need to be determined for tree breeders to determine whether they should be retained in breeding populations. This slide illustrates the results of a resequencing effort for 2 amplicons in the gene AGP6, which encodes the acid glucose 1-phosphatase enzyme in loblolly pine. Each amplicon, denoted here by the solid red bars near the top, covers a run of nucleotides between 415 and 560 bases in length. Haploid megagametophyte tissue from 1 seed from each of 20 trees was sequenced, and the results were aligned relative to each other. Color coding makes it easy to see the SNP sites. In total, there are 16 here, 7 in the first amplicon and 9 in the second. Individual tree genotypes have been arrayed in order, with those sharing a common set of SNP genotypes grouped together. The term haplotype refers to these common SNP sets. The haplotype is a powerful way to categorize and evaluate genetic variation. In this example, 8 different haplotypes are detected in a sample of 20 trees. Conifers enjoy a biology that makes direct detection of haplotypes possible. For most diploid organisms, inferring the haplotype is a technically and statistically difficult task. New sequencing technologies have made acquisition of DNA sequence relatively inexpensive and easy to acquire for virtually any species of plant or animal. De novo sequencing is a logical choice for many SNP discovery applications. The strategy chosen to acquire new sequence will be influenced by project objectives and goals, naturally. Things to consider when following this approach include what DNA source will be used, genomic DNA or transcribed DNA, that is, cDNA from mRNA. What population will be sampled? SNP discovery can be made by sampling DNA from a single diploid individual, but only for those loci for which the individual is heterozygous. As seen in an earlier slide, DNA pooling from a large number of individuals may be necessary to obtain evidence of most of the SNPs in a population, though this does not readily or accurately yield information on allele frequencies. Finally, choice of sequencing platform must be considered. Let's look briefly at DNA source and sequencing platform. A project's objective will directly influence choice of which DNA source is used for sequencing. If the primary purpose of the project is to sequence the genome, the obvious choice is genomic DNA. SNP detection is a byproduct of the sequence obtained. If the goal is to identify genetic variation that may be associated with phenotypic or environmental variation, a logical source would be the portion of the genome that codes for expressed genes, that is, ESTs. Gene-based SNPs have obvious evolutionary relevance, but there are some drawbacks to EST-based data. Consider the following points. Genes of interest may be missing from your messenger RNA sample. It may be difficult to obtain adequate sequencing depth for genes with low expression, that is, rare messages. SNPs and promoters, enhancers, introns, and intergenic regions will likely be missed, and the presence of related non-allelic sequences from multiple members of gene families will confound sequence consensus. The Sanger sequencing method requires a single-stranded DNA template, a DNA primer, a DNA polymerase, normal deoxynucleotide phosphates, DNTPs, and modified nucleotides, dideoxy-NTPs, that terminate DNA strand elongation. These ddNTPs are radioactively or fluorescently labeled for detection in automated sequencing machines. The DNA sample is divided into 4 separate sequencing reactions containing all 4 of the standard deoxynucleotides and the DNA polymerase. To each reaction is added only one of the 4 ddNTPs, that is, ddATP, ddGTP, ddCTP, or ddTTP, which are the chain-terminating nucleotides lacking a 3' OH group required for the formation of a phosphodiester bond between 2 nucleotides, thus terminating DNA strand extension and resulting in DNA fragments of varying length. The newly synthesized and labeled DNA fragments are heat denatured and separated by size, with a resolution of just 1 nucleotide by gel electrophoresis on a denaturing gel, which each of the 4 reactions run in one of 4 individual lanes. That is, lanes A, T, G, C. The DNA bands are then visualized by autoradiography or UV light, and the DNA sequence can be directly read off the X-ray film or gel image. The relative positions of the different bands among the 4 lanes are then used to read, from bottom to top, the DNA sequence. Sequencing platforms have evolved rapidly in recent years. The gold standard of sequencing technologies for years was based on Sanger's chain termination approach, as represented by a series of vendors' products like the ABI 3730. The advantages of Sanger methods were long read lengths, that is, greater than 750 bases at a time, and accuracy of base calls. A trio of second-generation sequencing platforms entered the market in 2007 and have enjoyed rapid improvements since then. They vary in cost and read length, but overall produce copious sequence at a fraction of the cost and time required for Sanger sequencing. Today, third-generation sequencing technologies that rely on single-molecule detection solutions are nearing market and promise to once again push the boundaries of sequence production. DNA storage and computing power for data processing are now technical and financial hurdles that need to be addressed to accumulate this flood of genetic data. Collectively, the newer technologies are referred to as next-generation sequencers, or NGS. This figure simply illustrates the remarkable trends in sequencing and data storage capacity with the concomitant declines in cost per unit of production. In short, the technology to sequence even the largest genomes, including conifers, at reasonable cost and precision now exists. By 2013, it is assumed reasonable to assume that hundreds of thousands of SNPs distributed throughout the genome will have been identified for each of several conifer species, providing the genetic reagents to implement marker-informed breeding, or MIB, in a meaningful manner. The example illustrated here reflects the pace of sequence acquisition and SNP detection now possible for virtually any species of interest. Of course, it does not necessarily reflect all elements of such a study. For instance, there was considerable time and resource invested in creating the complementary DNA libraries, and developing the pipeline for data processing required considerable investment in time. Regardless, more than 15,000 potential SNPs were generated in a very short time. Of these, 1,536 putative SNPs were scored for their potential conversion to usable markers and were submitted to genotyping on a high-throughput Illumina platform. From this, over 1,000 markers were identified and placed on a genetic map within months. Because of the rather sophisticated nature of current genomic tools, most scientists and new graduate students have become rather distanced from the lab. Most data acquisition, from construction of libraries and DNA templates to sequencing, are now done by contract and very high throughput vendors. In turn, investigators today are required to be experts in the field of bioinformatics, the handling, processing, and interpretation of the voluminous genetic data being produced, evaluating the millions, perhaps billions, of base calls generated for accuracy, aligning like sequences, and assembling a consensus sequence takes powerful software. These tools are constantly evolving. Each new sequencing technology poses a unique set of interpretational issues that requires new approaches to data processing. The sequence assembly problem seems relatively simple. Construction of a DNA sequence that is composed of all component sequence reads. This is generally true if the data did not contain sequencing errors, polymorphisms, and long repetitive, that is, non-unique, regions. These challenges, as well as the massive amount of sequence now generated from next-generation sequencing technologies, provide the foundation on which various sequence alignment or assembly algorithms are developed. An array of Unix-based software tools, largely developed by scientists at the University of Washington in Seattle, have been built and continuously modified over the years to process DNA sequence generated by Sanger sequencing technology. They are intended to be used in a pipeline that handles large data sets in a high-throughput manner. The FRED application accepts a DNA sequence chromatogram input file, such as .abi or .scf, and makes nucleotide base calls with assigned quality scores. FRAP takes this output, seeks sequence reads that appear to be covering the same chromosomal regions, and lists them as contigs. FRAP is an assembler and creates a consensus sequence with an overall quality score. CONSED provides a graphical alignment of representation of the contigs, thus allowing for user visualization and editing of the data. It allows for manual operations such as 1. Adding reads to an assembly, 2. Annotating reference sequence, 3. Reviewing sequence quality and genotypes, and 4. Identifying potential allele-specific or non-unique amplification, or pseudogenes. PolyFred is a program that compares fluorescence-based sequences across traces obtained from different individuals to identify sites for SNPs. The PolyBase application provides another quality check of the SNP calls. Though the various next-generation sequencing technologies vary in how they acquire sequence information, they all follow a common flowchart from DNA preparation to data processing. Most sequencing vendors provide their own analytical software, but a plethora of alternatives have exploded on the scene seeking to find improved ways to process the flood of data. Literally dozens of alternative programs exist for assembly and SNP detection applications. The biggest challenge facing data processing for next-generation sequencing rests with the relatively short read lengths they produce. Short reads make de novo sequencing and assembly difficult and computationally intensive. Consequently, Next-generation sequencing technologies were initially used for resequencing applications. However, clever strategies and improving read lengths have increased their utility. The biggest problem with short-read sequences from next-generation sequencing technologies such as Illumina is their inability to map uniquely back to the genome or transcriptome being sequenced. This technology is, however, very desirable because of the great abundance of sequence that can be generated in a short amount of time for very little money. To deal with the downstream problem of alignment and SNP discovery, short reads are now being paired with a small amount of larger read technologies such as 454. This next-generation sequencing technique produces a more substantial backbone for alignment purposes. Another technique widely used is paired end sequencing, where the short reads are sequenced from both ends, often leading to better success at mapping them back uniquely. This is especially important when genomes or transcriptomes are being assembled de novo. High-throughput SNP detection software may yield literally thousands of putative markers, not all of which are real. Errors in sequencing, alignment, or assembly may introduce false positive SNP calls that vary from 0.5% to 5% or higher. Furthermore, in very large, complex genomes, such as those found in conifers, an added concern is that sequence reads that are aligned may represent two or more chromosomal regions. This occurs because genes often represent large gene families that have developed over time through gene duplication. As a consequence, the SNPs detected may not be truly allelic. The classical approach to validating markers is to determine if the alternative alleles segregate in a Mendelian manner. With conifers, this could be done easily by Sanger resequencing for 4 to 8 megagametophytes from one or more trees for the sequence in question. Such an approach works well for modest numbers of markers, but would be prohibitively expensive and time-consuming for applications that seek to use thousands of markers. In such cases, it may pay to simply validate SNPs during a high-throughput genotyping exercise that included segregating progeny in the population being genotyped. Only those markers that behave in a Mendelian manner are used. Genotyping platforms for SNPs are many and varied, each with characteristics that make them more or less attractive depending on the application. As noted earlier in this module, factors to consider in choosing a platform basically focus on cost and accuracy of allele calls. Markers have had, and will continue to have, important roles in our understanding of genetic principles and applied genetics. The phenomenal increases in marker discovery have fostered strategies for new marker applications in all organisms, including forest trees. While Allozymes and microsatellites will continue to find applications in forestry, the SNP will likely be the marker of choice for most genomic-scale applications for the foreseeable future. Marker-informed breeding will become a routine and cost-effective complement to traditional tree breeding within the next 5 years, by about 2015 or so.